All right, let's get going. Thanks for coming along. So this is the second of our new seminar series designed at showcasing what the faculty does. And today's presenter is Professor Jim He. And he's from the School of Agriculture and Food, and he's going to talk about opening the black box of soil microbial communities. Let me say a few words about Jim. He received his PhD in molecular ecology from Griffith University here in Australia in 2005. And since then, he's built an eminent international reputation for working in the field of soil microbiology and nitrogen cycling. To go along with his eminent international reputation, he has an outstanding publication track record with over 185 peer-reviewed international journal publications. He's been a leading researcher in the group of molecular soil ecology at the Chinese Academy of Sciences in Beijing since 2005, and here at the University of Melbourne since 2013. Jim's led a number of large research projects, including ALC discovery and linkage projects, and he's listed as a highly cited researcher by Clarivate Analytics, which goes into establishing the esteem of the university and all of the metrics that go into determining university rankings. So there are not very many members of the university who are highly cited researchers. Jim's one of them. He's also a member of the ALC College of Experts. Please join me in welcoming Jim to give this seminar.
Okay, thank you, John, for your introduction. And it's my great pleasure to give this seminar. Actually, I gave a faculty or school-wide seminar when I was interviewed some 5 years ago. So it's a very good opportunity to give an overview of our research and also some challenges in this research field. So you can see my title is Molecular Soil Ecology. So Professor of Molecular Ecology. Many people ask me, what is this title? Actually, we say soil ecology certainly is understand the organism and with the environment in the soil ecosystem. But the molecular basically is an approach. So we try to use in the molecular technology to understand the relationships of organisms with the biotic and also abiotic environment in the soil ecosystem. So basically this is the meaning. So what is soil? This is a very general introduction. So soil is a very thin skin over the land. I often, when I give a lecture, I often said that. So what's the texture of your skin? If it's soft, is coarse, or this decide how the soil is fertile and also the soil productivity. So how can we take care of our skin? It's just like we need to take care of our soil. So it's important. The skin is important, and also is a mixture of minerals, organic matter, organisms, water, and air. So previously we studied the soil, more focused on the physical and chemical aspects. But now, actually, we put more emphasis on the soil organism, particularly microorganisms. And also certainly is a precious resource to support our human being, help to produce food and fiber. And the soil is alive. Why is it alive? Because there are so many organisms living in the soil system. So the soil contains one of the most diverse group of living organisms on our earth. So, and a single gram of soil can contain, we say, billions of microorganisms. So you can imagine how billion is big. And also, bacterial biomass can be 1 to 2 tons per hectare in the surface soil. So although we cannot see the microorganisms use our naked eye, but actually all those organisms are buried in the soil, and they drive the material cycling and energy transfer in the soil ecosystem. So even in the quantity, they are very big mass. And this is basically the composition of soil organisms. It includes, probably you know, like those fungi, bacterial, nematodes. There are different organisms in the soil ecosystem. And this is a slide I often use. So how many organisms or how many bacterial and fungi in the soil? So this is our S, and our population now is 7.5 billion. So a handful of soil, just like, handful of soil, just like the population, the organisms in the soil is just like our population on Earth. So a handful of soil can contain billions of organisms. So it's a huge amount. If give you some general idea on mass, biomass. So the biomass of organic microbes carbon is similar to the plants. You can see so many green plants on Earth, so many big trees, but in terms of carbon, they are similar in mass. In terms of nitrogen and potash, microbial nitrogen and phosphorus is even 10 times more than the nitrogen and potash, phosphorus in the plants. So you can imagine how big a mass, how big a diversity of organisms, particularly microorganisms in the soil. So we often see the soil is soil microbes are black box or is a black box. Why? Because most of them are not culturable. Because the conventional research or traditional research, we need to isolate the microbes. We isolate them, do the pure culture, and then we study the microbes and their functions. But actually, most microbes in the soil are not culturable. So only about 1% of soil microbes are culturable. So we often treat the soil microbes as a black box, Some years ago, even in the science, there is a special issue. It's called soils, the final frontier, because the soil is so complicated. Even just consider the organisms in the soil, how they communicate, how they involve in the elemental transformation or the nutrient cycling or the organic compound decomposition. So if you want to know who are there, how many are there, and what are they doing, or their functions in the soil ecosystem, is so difficult. So we need to have a very robust approach to study the soil microbes. So this is just beneath our feet. There are so many organisms, but what method we can use to study those so diverse organisms in the soil ecosystem? So certainly, there are culture-dependent methods. We can culture those microbes in the soil. We inoculate the soil suspension into the petri dishes and using different media, and then isolate those microbes, and then we can study the composition, the functions. But because there are so many organisms, microbes in the soil, so we call the culture-dependent method are biased. They are selectively culture those microbes. If we want to do a comprehensive study of the soil microbial communities or organisms, we need to use some other method like microbial biomass, we can weigh how big a mass of the organism have in the soil. As I mentioned, 1 hectare of soil can have several tons of microbial biomass. And also the biolog plates, this is depending on the culture method, and then we use different, for example, biomass. This is PLFA, but mostly molecular approach based on the microbial DNA and RNA, just like our human beings. Also, we cannot culture the microbes, but we can extract the biomass and then to identify the composition. Okay, this is basically the culture-dependent method. This is widely used for over a century. And in 1980s of last century, the molecular technology developed so quickly, and now everyone knows, we can quickly sequence, for example, the human genome and also plant microbiome, soil microbiome. So this is basically the simplified approach of molecular method. So usually we go to the field to collect some samples, or we can have sediments, water, or soil, or any, like we collect the manure in the feed lot. So we collect some sample, and then we need to extract the microbial DNA. So how can we extract it? So we need to crush the bacterial or the microbial cells. And then actually this is using chemical method to isolate the DNA from the matrix. So when I did my PhD previously, we don't have extraction. Now very easy to use the soil DNA extraction case. So we're using the chemical to isolate the microbial DNA from the soil system. After we got the DNA, because soil is a so complex matrix, some organic compounds can be co-extracted with the DNA, and we need to do some purification. And when we got we call the high quality DNA, we can do the further analysis, include PCR. PCR is actually we amplify the DNA. For example, the DNA is not so enough for further analysis. We can do many copies of the same DNA, and then we furtherly doing the qPCR to quantify how many copies or gene copies of the DNA have. And also we can do the cloning sequence and then identify the microbes in the soil. And this is we call the collectively fingerprinting method, just like a fingerprint. Everyone have your own fingerprint, and the different soil have the fingerprinting profiles. Just like a diagram of your print or of your fingerprint. So this is this method are based on the gel electrophoresis. So different DNA can move with different speed, and also different size of DNA, and the DNA composition will differentiate on the gel. So in this way, we can see the fingerprinting profile of each soil. But now we have a much more approach. It's basically multi-omics method. As you know, metagenomics. We can quickly get the DNA. Sometimes we can do some amplification and then sequence the microbial community and then do further analysis. So this is basically a simplified approach of molecular technology. So what this technology can tell us, or what knowledge we can get from this kind of research? Basically, so who are there? So in this soil ecosystem, or with different land use or different crops, different altitude, or with different land change, and a different situation. So who are there? How many are there? Why they are there? This is basically the ecology. What energy source they can get? What are they doing? What the role are they playing? Particularly in the agricultural system, they are more involved in the organic matter decomposition, nutrients cycling. This is more related to our field. And also, how will they change? How will they change? Yeah, we often mention about global change. How the global change will result in the microbial community change. And also, the function of microbes will change. So basically, this is my research field. So if we, firstly, we need to get the microbial DNA. So we quite often go to the field to collect the soil samples, and then we're using this is a fast prep to crush the soil microbial cell, and then we can isolate the DNA. So how can we get the, we call it high quality, size, bigger size, and also pure DNA. So I gave an example. This is what I did during my PhD. So this is, for example, a forest system. There are different rotations. This is a natural forest. This is a hoop pine, first rotation and second rotation. Because the soil is a very complicated matrix, they include, we call the humic compounds. So there are a lot of organic matter in the soil, and those humic compounds can be co-extracted with DNA. So we need to develop some method to improve the soil DNA extraction efficiency and also the purity. So I developed some method. We can wash the DNA to remove those dissolvable organic compounds, and then we do the DNA extraction. This will greatly improve the DNA extraction efficiency. This is just a very simple example. And then if we got the DNA, we can amplify the DNA. So make many copies, usually some billions of copies of the DNA. And then if we got enough pieces of DNA, we can carry out further research. For example, if we want to know, who are involved in the PCB degradation, because PCB is quite used, often used pesticides and disinfectant, but actually in the environment, it is very difficult to degrade. We don't know any specific microbial species are involved in the degradation of PCB. So we can do an experiment. For example, this is a control, and then in the soil we add the PCB, and then we run the gel. This is we call the DGG. The gel is a type of fingerprinting method. So we can see how the microbial band will change with time. So after some time, after 49 days, 63 days, and then we can see the band. For the PCB, actually, after we add the PCB to the soil incubation, some bands are quite strong. It means those microbes are involved in the degradation of PCB. If not involved in the degradation, at least PCB are not toxic or to depress the growth of those microbial community. And then we cut the band and do the sequence, and then we can know which microbial community are involved in the PCB degradation. Furtherly, if we are not sure, we can use we call it stable isotope probing. We labeling the PCB using carbon-13, So if the carbon-13 can incorporate it to the microbial DNA, those microbes must involve in the degradation. of PCP. So in this way, we can track which microbes are involved in the PCP degradation. Okay, so this is just a general introduction of the microbial or molecular method we are using. Now I give you some examples of our research. So the first case study is soil microbial distribution patterns. We call the biogeography and the temporal succession. So how the microbes are distributed on the Earth, all the factors are governing the microbial distribution, or all the theories can predict the microbial distribution and also the succession with the time. So as you know, for example, this is the vegetation tables. Yeah, the plants have some distribution pattern or geographic distribution patterns, and also like these small penguins, they have some specific distribution area. So can we now have some indicator of microbes on map? This is generally called biogeography. So if we can identify those microbes in different areas and what factors or what environmental conditions are governing the distribution of the microbial community. So this is a microbial biogeography. Can we put the microbes on the map? So the basic question is, what is the distribution pattern of soil microbes? And then what factors are driving the pattern? This is basically more about the neutral theory or some different factors are controlling the microbial distribution. And how do they respond to environmental factors? If we can develop some model to predict the microbes and also the functions succession, for example, with increasing temperature or with the increased rainfall, how the microbes and the function will evolve, will change. So this is some basic questions. So actually in the world, we have, many people have done the microbial distribution like this is in UK, published 2011. This is in France. This is in China. We did some years ago. About this is archaea, this is bacterial, and also this includes bacterial and fungi distribution in different areas. So I give you an example I did in China. So this is basically the map of China. So we collect samples from the forest ecosystem because forest ecosystem has less human disturbance. So it more represents the natural conditions. So we from very south to very north to collect the soil samples, and also we need to identify the above ground plants. So we collect the samples. We need to have quadrants and collect the plants and also trees and also soils. And this is collect many different soil samples, and then we do the DNA extraction and we sequence it and do we call the bioinformative analysis. Analyze the relationship of microbial community with the environment. This environment includes biotic environment. The relationship with plants. So this is basically the tree composition across latitude biomes, because from north to south is some 4,000 kilometers away. So certainly the plants are quite different. This is geographic distribution, and also the understory helps plants composition and the grass, how will change. And then the microbes, how the microbes will change. I will not go to the details. And this is in different levels. The previous one is in the phylum level, and this is the class level. And then we study the relationship of microbes with, for example, this is a tree with the increase in latitude. the tree diversity decrease. This is herbs, grass has this form. This is microbes. Certainly, you can see just from this graph, you can see there are very similar pattern of microbes diversity with grass diversity. Certainly, we can do some further analysis to understand the altered environment driving this pattern, driving these patterns. So for the soil microbes, herbs and also soil pH play important role in determining the microbial distribution or the bacterial distribution in this soil. If we do some synthesis, incorporate the previous data to, for example, together with America, Australia, China, New South Wales, we can identify which factors are driving this is a very large scale distribution of microbial community. So actually this is a paleoclimate because the soil development is largely driven by some paleoclimate, certainly temperature, vegetation, many other factors. So we found paleoclimate. This is about 10 to 20,000 years ago. The climate played a very important role in determining the microbial distribution pattern. So this paper, in 2017, we published in the Nature, Ecology and Evolution. So this is a very large-scale studies. So in China, we collect a sample from very north to very south, very east to very west, even to the Tibetan Plateau or to some glacier. So we can collect those samples and study the microbial distribution. So we have, I established we call the SSB archive. This project, we collect several thousand samples. We study the microbial composition and their relationship to the environment. So we collect the samples. This is, I suppose, is a very good way. We have each tube, we store 50 grams of basically it's a freezer-dried soil. And also we extract the microbial DNA and put it in the very tiny tubes and store in the fridge. Because if, for example, quite often after 5 years, 10 years, we can go to some sites like a glacier retreat area and to collect the sample and to see the change of microbial community. So this is called temporal evolution or temporal succession of microbial community. So last month, we also did a major sampling campaign in particularly east to central Australia. Hanwei led the team and drive about 10,000 kilometers, 10,000 kilometers to collect the soil, grass, and tree leaves samples. So we will do some further studies of soil microbiome and their relationship to the plant microbiome. Particularly, we try to understand the indigenous plants or habits, the soil microbiome and their relationship with the plant microbiome. And this will have some meaning, certainly it's a resource distribution pattern, and also the drive factors, and also cycle or co-evolution of soil and the plant microbiomes. At the moment, they are working on the antimicrobial resistance genes, but for more important aspects, how the plant and the soil microbes co-evolve to adapt it to the very deficiency situation of, particularly for phosphorus, because Australian soil has a deficiency of phosphorus, and also in many areas there are deficiency of nitrogen and also some other elements. So in this way, we can understand why those indigenous microbes, plants can survive in those very tough situations. So this information can be transferred to the breeding, synthetic microbial ecology, and even for the soil remediation. So there are some very significant meaning. And in this field, we have done a lot of work in China, and also now 5 years ago I moved to Australia. We will work on the microbial biogeography. So in this field, this is the ISME, is the International Society of Microbial Ecology. This journal, this is the top journal in our field. It's microbial ecology. We published many papers in this journal, and also the meeting next year in Cape Town. I'm the chair of we call the global and continental biogeography. So, and also I quite often invited to give some, invited keynote speak presentation in this field. So this is, I think, it's important to understand the microbial distribution at the national or even the continental or the global scales. Okay. So this is a very big scale or the overview of microbial distribution. But you must, to quite want to know, because microbes is very tiny, very, very small size. In much smaller scale, how the microbes can play the role. So I will show you another example. It's about called the case study example 2 about nitrogen cycling. So because we have an agricultural background, nitrogen is probably the most important element or nutrients for the agricultural ecosystem. And nitrogen cycling in the ecosystem are driven by the microbes from nitrogen fixation, nitrification, denitrification, and modification. All these processes are driven by microbes. So my research is more focused on the, we call the nitrification process. In this process, the microbes convert ammonia to nitrate, and then from nitrate to nitrite. This is the nitrification process. So usually we divide the microbes involved in the nitrification. They are carried out by two organisms. The first step is the microbes convert ammonia into nitrite, and then in another microbes, they convert oxidized nitrite to nitrate. And this nitrate, they can throw denitrification or throw leaching to influence the environment. So the nitrate can be leached to the groundwater, causes eutrophication, and also produce nitrous oxide as greenhouse gas. So we need to understand the microbial process and then develop approach to control this process. So it's essential to know which microbes are playing the role and in what way they are playing the role. And then we call it manipulate the environment to control those processes. So this is, traditionally, we think the nitrification are carried out by the bacteria we call the ammonia oxidizing bacteria. But those ammonia oxidizing bacteria are not growing well in acidic soil, in acidic conditions, because the optimal conditions is basically from 6.5 to the 8. So how the microbes play the role in the acidic condition. So we don't know how the nitrification occurs or happens in the acidic soil. But actually, in the acidic soil, for example, pH below 5, there are quite active nitrification. So who are playing the role in the acidic soil. We don't know. Until 2005 and 2006, 13 years ago, it was found in the marine system and also in the soil. There are many archaea. Archaea is another group of microbes. They can play some role in the nitrification. So we carried out a lot of research in this field. So this is in an acidic soil. This with a different fertilization treatment. So we have the treatment like this, no fertilizer. This is nitrogen and phosphorus, nitrogen and potash. We have different fertilization treatment. And this treatment results in the change of soil properties. For example, for the pH, with the nitrogen fertilizer application, pH drop several pH unit, like with only nitrogen is 3.7, but for the CK or like this is actually 5.8 or like this. And also this results in the change of, for example, nitrate concentration, and this is potential nitrification rates. So the nitrification capacity in the soil changed with different fertilization. So we want to understand what microbes are playing the role. So this dark color is AOA, ammonia oxidizing archaea, and this white one is ammonia oxidizing bacteria. You can very easy to see the dark color is higher than the white color. And also AOA and AOB, and also this is the potential nitrification rates. This is the potential nitrification rates keep change in different soil. And also the composition of the AOA changed. For example, this is with nitrogen and phosphorus, nitrogen, potash, or the nitrogen application composition of AOA changed dramatically. And then in another soil, so this is a soil we call the alkaline sandy loam. So this is also the dark color is AOA, and the gray color is AOB. And this dark color is much higher than this gray color. The AOA is several times to several hundred times of AOB. But only AOB, bacteria, has a positive relation with potential nitrification rates. But AOA not. Actually, so in this alkaline soil, only AOB play predominant role in these alkaline sandy loams. Also, AOA are more abundant than AOB. So actually, in different soils, the K players are very much different. So we want to know in each soil who are playing the role. So previous these analyses are based on the statistical analysis, but we need to have more strong evidence. So we developed we call the carbon-13 stable isotope probing. We can label the substrate. Here the substrate is carbon-13 or carbon-12 carbon dioxide, because AOA and AOB are autotrophs. When they oxidize ammonia into nitrate, they need to fix carbon dioxide. So we do the incubation. So if the microbes are playing the role, the DNA, the carbon-13 will be incorporated into the DNA. And the carbon-13 DNA is heavier than carbon-12 DNA. So we're using the ultracentrifugation, to differentiate carbon-12 and carbon-13 DNA. So in this way, we can identify which microbes are involved in the nitrification. So this is basically the mechanism. So we incubate the microbes with carbon-13 labeled carbon dioxide to see if this carbon-13 can be incorporated to the microbial DNA. So we extract the DNA, and to measure this is a buoyant density. So if you can say, after 2 weeks or 4 weeks incubation, we extract the DNA. This is for AOA. The DNA shifted to the heavier group. It is heavier than the carbon-12 DNA. It means those AOA play the role in the nitrification. But this is for the AOB. The peak are still in this area. And then we can further identify which, for example, which AOA play the role. This is the AOB. There is no band in the heavier group. This is the AOA. After 2 weeks or 4 weeks incubation, there are many bands. Those microbes, those archaea, play the role in the nitrification. So we developed this method and published in the PNAS in 2010. So this is a very good method to track which microbes are playing their role. So we extend this method to many acidic soil. All these soil are pH 4.5, below 4.5. So we can find, ammonia oxidizing archaea have more important role than ammonia oxidizing bacteria in the strongly acidic soil. So we did many research in this field, even using several hundred of samples, and demonstrate in the acidic soil, the active player of nitrification are archaea instead of bacteria. So this is a very good example. How can we use the molecular technology to target the specific microbial community and also understand their functions? And recently, there are another group of microbes we call the common mox. This is just discovered 2 or 3 years ago. This is a new group of microbes involved in the nitrification. Previously, I mentioned, nitrification is a 2-step process. So the process are driven by 2 separate microorganisms. But actually now it's found there is a common mox. It's called complete nitrification. The 2-step nitrification can be complete just in one organism by one-step reaction. So we did a lot of research in this field. So this is several hundred of samples. We screening, what microbes exist in those soils. So this is AOA. I mentioned ammonia oxidizing archaea. And then this is AOB, this is AOA, this is AOB, and then this is common mox. Common mox can be divided clade A and clade B. This is a different group. So you can see, actually, there are quite a high common mox in those soils. So at the moment, I have several students working in this field, and also, 2 weeks ago, actually we got the first evidence of common mox play active role in nitrification in the eutrophic agricultural soils in Australia. So we submitted it to the Nature, but I don't know if it will be successful. But certainly this is important work to firstly give the evidence of those common mox widely existed in the Australian soils, and also they may play the important role. So this kind of research will provide the basis for the development of inhibitors. We're working on the nitrification inhibitors. So what kind of, how this research contribute to the agriculture management or the nutrients management, because or more than 50% applied nitrogen can be lost into the environment. This causes a lot of environmental detrimental problems, greenhouse gas emission, also eutrophication, and the nitrification process accelerates soil acidification. So we need to develop measures to manipulate the nitrification microbes. We need to understand the microbes and then develop measures to control, or we can call them manipulate or adjust the microbial process. In this way, we can have our findings to serve to the agriculture field. So in this field, I have one ARC DP project and also an LP project working on the inhibitors. So this basically shows you how the basic research help you to establish your research capacity, and also the possible use can link to, through, for example, a linkage project to develop engagement with industry partners. So in this field, I have many highly cited papers. Like I mentioned, this 2007 papers published this some time ago, cited about 600 times. We have the publications in environmental microbiology, in Nature Geoscience, and certainly in the PNAS, ISME journal. All those papers are highly cited, and also I'm invited to have some, I suppose it's important review papers. We summarize the progress in the field and also give the perspective in the future. So in this way, we can certainly, this is very important for my, for example, ARC discovery application or like this. And also, certainly highly cited researcher in this field. So some take-home message. So I give a very general overview of how the molecular technology help us to understand the microbial distribution and also microbial functions involved in the nutrient cycling, and also how can we use it to solve some agricultural and even environmental problem. So basically, I hope you can remember there are huge or tremendously diverse microorganisms in the soil ecosystem. So how many? There are billions. There are billions of microbes in even just 1 gram of soil. And also, molecular biotechnology can be widely used to understand the soil microbial diversity and functions. So they are so widely used, now a lot of research on the human microbiome, and even use the human microbiome to develop some medicine, help to treat the human disease or like this. For the soil, it's the same, because in the soil there are so many we call the soil-borne disease. This is mainly caused by some pathogens in the soil. So how can we develop an approach to control the soil-borne disease and improve productivity? And also, how can we, through understanding the indigenous plants microbiome, to help improve, for example, plant breeding or like this in the future? And also the basic procedures, as I mentioned, we can collect the sample, extract the DNA, do the PCR, do the fingerprinting, and then to sequence the microbes, and then link the microbial composition and the diversity to the environment. And this will help us to understand the relationship of microbes and the environment. Why we want to do this? Because we need to control some microbial process to serve our agriculture and the environment, okay? So the soil microbial ecology study play an essential role in understanding soil process and soil sustainable management. Thank you.
Thank you, Jim. I think we have time for a few questions. questions? While you're thinking of the question, may I ask the first one? So, Jim, how stable is the microbial population of, let's say, the soil outside this lecture theatre?
Yeah.
Does it change? You talked about pH being a major driver of the microbial community, but does it change with summer and winter, the temperature or the amount of rainfall? Or given that there's 7 billion or more bacteria in one handful of soil, are there just so many that it's sort of constant, whether it rains or it's hot? Or
John, as a virologist, professor of virologist, gave a very important question. Yeah, actually, we think the microbes will keep change in the soil. Yeah, certainly, yes. The answer is yes, they keep change. For example, after rain or after temperature change from winter to summer, or just from different crop system, or after fertilization, the microbes keep change. But we call the co-microbiome. So there are, for the soil microbial community, there are a part of composition are very stable. It's much stabler than we think. So why they are so stable? Those microbes, because they are involved in the basic functioning. So microbes basically can divide it to two parts. Basically, there are a lot of research. Some groups are very stable in the soil ecosystem. Some are keep change with the environment. So it's depending on the environmental conditions. And now many research are more focused on the more stable microbial community because they are playing basically more functioning.
Questions? Yeah, please.
It's probably a very silly question, but how do you adjust your collection technique for different soil environments? Because I imagine the depth at which you sample must make a big difference, but also if you're sampling in a rainforest where there's a huge layer of organic matter versus a dry, arid environment, so how do you standardise with that so you're comparing like with like?
So for the soil microbial study, the scales are so important. If you are from national scale or continental scale, or even in the paddock scales, certainly different, for example, soil type or different paddock, even the different depths of soil, the microbes will change. But it's very much depending on your scientific question. For example, in the agriculture land, we usually just collect the soils from 0 to 20 centimeters or 15 centimeters, because in this layer or in the horizon of soil, microbes are much more active and also involved in the order process. So we need to carry out the research at here or based on your questions, scientific questions. So we carried out a lot of studies. So how the microbes will evolve with, for example, the altitude from 100 meters to, we even worked to the Mount Everest, 6,000 kilometers, and also, from very dry area to very wet area, or like this. So if you want to study, we call it ecological theory, and understand all the relationship of microbial distribution with the environment, we need to use a gradient. For example, the rainfall gradient or fertilizer gradient. So have one factors variable with the environment, and keep other factors fixed, and then to study how the microbes will change. This is also important for the modeling, because now we think global change is a big issue. So how the microbes will evolve with the changed environmental conditions. So you can have a temperature change, rainfall change, or land use change, and then study identify the major factors or the key factors, and then develop some modeling. Yeah. Okay.
Did you have a question?
Yes, thanks, Jim. So many of your molecular methods don't differentiate between live and dead bacteria. Do you find this probably wouldn't be such a big issue when you're looking at biogeographical distribution of microbes, but probably more important in agricultural systems where the pH is shifting and the management strategies would change the number of live bacteria. Do you take this into account in any of your study?
Yes, yeah. So if you want to work on some specific functions or the alive microbial community, you can use RNA instead of DNA. But there are a lot of research in the soil system because RNA extraction is much more difficult than the DNA. And the research shows there are some positive relationship between RNA and DNA. So for the active group research, for example, I use the stable isotope or like this to target those active components. We can use in the RNA instead of DNA. But for the general environmental study, you can still use in DNA. Yeah. Okay.
Frank, did you have a question?
Jim, there's a lot of interest in antimicrobial resistance, humans and animals. What's the role of the soil microbial biomass as a reservoir for antimicrobial resistance, or as a source of antimicrobial resistance?
Yeah, that's important. Yeah, so I have a discovery project to understand how the antimicrobial resistance can be transferred from the feed node, basically from feed node to land, to the soil, if we apply the manure to the soil, and then from the soil to plants, we use the vegetable. So how the antimicrobial resistant genes move in this different matrix. So basically we using we using the model system to understand the transport or the transmission of antimicrobial resistant genes in the system. So we target the different genes, antimicrobial resistant genes, and study the quantity and also the composition change in different systems. Even for example, for the vegetable, we study the antimicrobial resistant genes in the rhizosphere soil, on the root surface, within the root, we call the endophyte microbial community, and also on the leaf surface, we call the phyllosphere microbial community, to study the linkage of antimicrobial resistance and understand what's the possible pathway of the movement of antimicrobial resistant genes in this system. For example, we're using basically mycorrhiza, we're using the fungal hyphae as a model system to target, to track the movement of the genes from, for example, from soil, outside the soil to inside the plants. So this is some fundamental understanding of the basically dissemination or the transport of antimicrobial resistant gene. But we don't know if there is real health risk if the antimicrobial resistant genes, for example, transported into the plants. Okay.
Okay, question over there.
Yeah.
I'm not a soil scientist. You drew the parallel between the human microbiome and the diversity here, and I'm curious, in terms of that last case study you were talking about with nitrification, you're essentially identifying very beneficial microbiomes. So in the future, are we going to think of microbiomes in the same way we think of other inputs like fertilizer, where we'll just have this package of very beneficial ones that will add to the soil in the same way that we do transplant?
Yeah, very good question. Actually, we do a lot of this kind of research. So in my group, we convert basically organic waste into fertilizer, do the composting, and then inoculate the beneficial microbes into the fertilizer, and then apply it to the field. In this way, they can improve the soil resistance to pathogens or to plant pathogens. So we call this bioorganic fertilizer. Okay, thank you.
Okay, last question and then we're going to finish.
Jim.
Yep.
So there's increasing concern about microplastics in the environment, including soil. So can you comment on the possible impacts of interactions to the soil microbiome?
This I talked many times. Any disturbance or any chemicals introduced to the soil will result in or induce the changes of microbial community. So for the microplastics, the research should focus on the microbes. They can break down or they can degrade the microplastics. Because microplastics is very small size. Actually, the physical impact to the soil is not like a plastic mulch, but they can play some role in soil physical and chemical characteristics. So the current, there are many research in this field. I don't think it's in a good direction of the research. Microplastics play important role in the water system, in the marine. They can end the fish to the animal, and this can further end the food chain. But for the plastics, microplastics, if you measure, if you study, if they can be uptaken by plants and further end the, for example, food chain, probably is more important. Okay.
On that note, I was reading an article the other day that said we all eat a credit card of microplastics a day. Is that what you were thinking about? Did you see that one? Yeah. Okay. Jim, thank you very much for that. Please join me in thanking Jim. Thank you.